Good afternoon, and it's great to be able to be here and make a contribution to this tribute to François, who's a great scientist and a colleague who I much enjoy the company of. I want to not spend so much time on data and think there'll be a little bit of data, but it's more about ideas and his influence on those ideas. And it's about François and the nexus. And you saw a lot about this morning. I think LEPSE is really leading the space in this area, mainly because of François' contribution. So what is the nexus? And to me, this is the way that I've been operating, and I have to obviously have a discussion with Bertrand after his initial remark, that it's the key role of interaction between experimentation and dynamic modelling in understanding and predicting crop adaptation, and doing both at the same time. So it's this loop. It's the loop between the mathematical world and the real world, where you're modelling, you're doing analysis and prediction, that's informing your experimentation, which is informing your modelling. Now, a lot of you, that's nothing too obvious, but to a lot of experimentalists, this is actually earth-shattering. But the nexus is really about doing these things simultaneously. So if you don't understand it, you can't model it. Often you get that from very experimentally minded people who say, I have to do more experiments, I don't understand it, you can't put this into a model yet because we don't know enough. To which the retort is, if you don't model it, you can't understand it. So you really have to think about dynamics to be able to capture the understanding that you're trying to get inside the system, which is a dynamic biological system. We can expand that out a bit and show the components inside here. So in the mathematical world, you've got these algorithms that enable you to predict key aspects of crop growth and development dynamics. And you can use that to do that list of things there. And all those things are particularly relevant when you come around through the analysis and prediction into the real world of either experimentation for understanding or for agronomy or for breeding, where you're trying to observe, understand key aspects from experiments. And so these are sort of the areas of work that you can get into in this loop called the nexus, okay? And the nexus is what I think is the critical thing to advance. And I was very impressed every time I came to LEPSE because you saw the progression of this thinking over time. And I can see it again now. It's fantastic. And so what's François' role in here? And so I think François has generated what I would call significant global influence on this space, certainly on me. And I only want to talk about two aspects because I won't have time to talk about any more. And I've got this sort of Tintin-esque view of François sort of looking into the world and with the vision, where's a big problem? I can solve it. I'm a detective. I can do anything. And so off he goes and finds the right answer for the right reason, the difference between cause and consequence, and dealing with issues of scale in biological systems, in the ecophysiological responses, norms of reaction, and the concept of meta-mechanisms. I just want to talk about those two things. There's lots of others, but that's all I've got time for. Mostly we do this sort of thing in a pub. And so I've got the pictures down the bottom here to prove it. The first set, Jean-François, it's here. This was a meeting in Rome at the Hilton for a GCP meeting, and the beers at that Hilton were way too expensive. So we took off down the road and we took François to the Guru Lounge, an obvious place to take François. And Scott Chapman was heavily involved in this, as always. And Scott also sent me this picture, which obviously we're talking about the nexus over a beer. What else could it be? Whoop, next one. So the first one, the right answer for the right reason, a beautiful, simple example from this great paper about modelling leaf expansion, specific leaf area. And I did say I won't have much data. So this is just a reduction in light for photosynthesis. This is saying it didn't cause much reduction in expansion rate, it changed specific leaf area. Now, you think, oh, that's pretty simple. But when you look at how people model this, they still get it wrong, okay? Here's what many models do, is they get a simulation, they take a specific leaf area, and they predict leaf expansion. Wrong. I mean, that was demonstrated, how long ago was that paper? Over 20 years ago. And it's just a beautiful piece of work. And some models do it the right way around. And it's actually, people say, well, so what? If I'm just trying to predict leaf area, both of these ways predicted about right. But the real point is you get the wrong links to the underpinning genetic architecture. So you've got to get the right answer for the right reason if you want to start linking models back to genetics. And that's a classic example of a beautiful piece of work that's sort of designed almost to say, hey, this is how you should model this. The other one is the meta-mechanisms, and I have to read a bit. People have already shown this fantastic paper of François. But there's a paragraph in there, I'm just going to read it. Modelling can simulate the responses of virtual plants carrying diverse combinations of alleles under different scenarios of abiotic stress. The main difficulty is mathematically expressing the genetic variability of responses to environmental conditions. Modelling via gene regulatory networks is not feasible for such complex systems, but plants can be modelled using response curves to environmental conditions that are meta-mechanisms at plant level. Each genotype is represented by a set of response parameters that are valid under a wide range of conditions. Absolutely beautiful paragraph. And that concept of meta-mechanisms has enabled the delivery of research across scale of biological organisation, certainly in models and I think in general. So it is an amazing advance, 2003. So this gave us the capacity to do all the things that have now happened. It gives you that ability simply to move across scale, all the phenotyping that flows from that, all the genotyping, and in models to link to phenotypic outcomes, predictions. So this concept of meta-mechanisms is really an amazing contribution. The example, again, has already been mentioned, the piece of work on leaf extension rate by Mathieu Raymond and François' group, to just show that leaf extension rate is essentially function of temperature and plant water status. Claude and others we added into that the connection of the same QTLs that affect leaf extension coefficients to anthesis silking interval. And so you get this trait by trait interaction going on across the genetics. With Corinne Chanou as a postdoc, we then captured all that into a sort of leaf size level model that we developed from the APSIM maze model, and you could then put the QTL in not only on the leaf elongation parameters but also on ASI, and you got that generated outcome of the same genomic regions giving you different yield outcomes in different environments because of these linkages, to leaf expansion but also affecting ASI, which has some negative consequences in some environments. So same genetic weight, G by E. And so you could get that stuff to emerge. Absolutely great stuff. There's another one that we did in the DROPS project, which I won't spend a lot of time on, but this was connecting this back with, I've obviously flipped something wrong there, connecting this back to silk extension rate as well. And so the work of Olivia Turk and Vincent Uri giving some detailed understanding of silk extension and how that was controlled by hydraulics, working with Charlie Messina, so we had Pioneer involved in this in the DROPS program. We were able to develop a model that linked the understanding of the hydraulics with the understanding of the timing of water stress through the crop and the availability of the plant sugar status and how that affected grain growth. So you could model essentially the number of grain, the size of the ear, and the grain abortion on that ear. And you could do it remarkably well. So we could model the different types of drought susceptible or drought tolerant genotypes by changing coefficients and generate these types of ear responses under different levels of stress at different times of crop development. So again, great idea, great concept of using these meta-mechanisms and how to connect them back to genetics. Beyond that, the meta-mechanisms you can think about, and I'd always sort of had this view of thinking about them more as adaptive strategies in a plant and how they connect with evolution. So it's more about what happens over time with either natural selection or just selection. And so these become central in looking at how you might manipulate the system genetically. And the way that we sort of wrote this review paper around this idea. François had been thinking around this for a degree of time as well, and this sort of came out of I think the iCropM meeting that was here. And again, I'll just read it. We propose that natural selection has constrained the large diversity of mechanisms into consistent strategies, so adaptive strategies, in such a way that not all combinations of mechanisms are possible. These constraints and resulting feedbacks result in integrative meta-mechanisms and result in robust and stable equations with heritable genotype-dependent parameters. And that's a sort of really neat way to think about evolution, because it's not like the selfish gene, it's the selfish strategy, okay? And it's the phenotype at the individual level that really is what natural selection is about, and this is how you get change at that level. So where can you take that? What we've been doing, and I'm very pleased to see it's not very different from what's looking like it's starting to happen here, is we're starting to use these sorts of models as gene to phenotype link functions. And so this is a review paper with Mark Cooper, where you take the traditional breeder's sort of gene to phenotype sort of approach, and you start to put a crop model in here, and you can, so you're doing sort of biological epistasis. And so you're bringing components of the genome into traits, and the dynamics in the model does much of the G by E for you. And you can go further and start linking it to networks, ultimately. So what we've done is we've hooked that up into our crop modelling platform, AppSim. We now have an AppSim whole genome prediction platform, and we're using that to play around with selection strategies about how you might go about enhancing genetic gain. So you can put that into future climates, you can put it into different traits and different backgrounds, and look at these component traits and how they interact over time. So the main thing here, and as I said, I think I was very impressed with Randall Wisser's talk, that I think he's sort of doing the same sort of stuff with Pierre and others. So I think this is sort of where the future is heading a little bit. With whole genome prediction, because of now the capabilities with genetics technologies, you know, your phenotyping may not be as relevant as it was, okay? But don't give up on the platforms, please. So the thing I want to say to just finish this is it's really the insights, those few insights from François' work that have really informed a lot of this development. And it wouldn't have been possible without that. So the contributions to me come from those insights and ideas that he promoted and discussed over many beers. So that's François and the Nexus. Thank you, François.